Uh, Hello. So before starting, I want to ask whether you guys are having any doubt regarding any lectures or regarding any assignment. Kalpana and Shinjini, whether you guys are there.
Yes, ma'am.
So you are having any doubt regarding any assignment or any
No, ma'am. No, ma'am.
okay. So hello everyone. I am Nilanjana, TA under Professor Himanshu Kumar, and in the course name host pathogen interaction immunology. So this is week 11. So now sir is teaching about bacterial infections and mycobacterium especially. So today I will just go through the same bacterial infection, and two people had some doubts which they posted in the portal. So I will discuss about the doubts also in the end of this session. So if anyone is having any doubt or they are having any kind of question regarding the lectures or any kind of assignments or regarding my lectures, then you can raise your hand or you can write in the comment box also in between. Uh, so today we are going to study about bacterial infections. So I hope you all know what a bacteria is. There are so many kinds of bacteria. in our environment. To begin with, I will start with the structure of a particular bacteria. Uh, like bacteria, height of a bacteria, like bacteria are very small. They are very much ubiquitous in nature. They can be found everywhere, but they are very small. We cannot see them in our naked eye. They can be seen under microscope. So, height of the bacteria can range from 3 to 4 micrometer. You can understand how much small that is. Then is a cell wall to protect it from the outside environment, harsh environment. Then again, like we are having a plasma membrane, the bacteria also has a cell membrane. Uh, there is a capsule, that is a polysaccharide structure, some of which may be shaped or dissolved in surroundings. Be DNA, different forms of DNA. Mostly the bacterial DNA are circular in nature, which we call as plasmids. There can be one or more circular DNA plasmids, and they can code for toxin production or they can code for antibiotic resistance. As we all know that many bacteria now have grown resistant to antibiotics, so they encode some genes from within, like which they have in their structure, and that those genes only give resistance to the particular antibiotics. Here we can see this is a circular DNA genome, and there can be different kinds of structures present in the bacteria. Like example here, pili. It can be more than one type. It helps in adherence of the bacteria to the host cell surface. So here this rope-like structures you can see, and also there can be flagella, which helps in the motility of the bacteria. So it can move with the help of this flagella, and with the help of pili, it can get attached to the surface of the host cell. So this is just a basic structure of bacteria. It varies from one bacteria to another. So it can enter through our body through a number of routes. Example, it can enter through our respiratory system. It can enter through our gastrointestinal system. It can enter through our urogenital tracts also. Or they can also get access to the breaks in our membranes or the skin. I have told previously also, whenever there is a break in our skin or like that is the most vulnerable spot where a pathogen can enter into our body. Any kind of wound or any kind of breaks in our mucous membrane or even in skin also, any kind of wound is there, that then there it is more likely that it can get infected, because pathogens can enter like enter easily, and skin is one of the most like important barrier for this pathogen. But if there is a break in the skin or any kind of membrane, these pathogens can easily enter. Slide is not changing. Okay. Is it changing now? Kalpana? Okay. So bacteria can enter through our different kinds of routes, and as you all know that our environment is full of bacteria. So even if there is a small chance, any kind of bacteria can enter into our body, and there are different like depending on the number of organisms entering and their virulence, our different levels of host defense are in there. Like whenever there is an entry of the bacteria or any kind of pathogen in our body, we have a different kinds of host defense against different kinds of pathogens. And if the size and the virulence are both low, then the tissue phagocytes may be able to eliminate the bacteria via non-specific innate defenses. So it need not like uh, activate the more robust immune response if tissue phagocytes are already there, which are circulating in our tissue, or there are cells which are circulating in our circulatory system like the blood. So if there is a minute, like some virulence is low, or the size is also low, then the tissue phagocytes are enough to eliminate the bacteria via non-specific innate defenses. If the virulence is high, or if the size of the organism is also high, then it requires more robust immune responses like antigen specific adaptive immune response. In some instances, disease symptoms are not caused by the pathogen itself but by the immune response, as I already told you earlier also. It is not always the pathogen that is creating a kind of symptoms, or kind of what to say, like bad things which are happening to us, like inflammation or something. It is but the immune response that is acting against the pathogen, which is actually creating that response. Like for example, there can be overproduction of cytokines, there can be non-discrimination and systemic expression of various proteins, there can be bacterial septic shock, which is one of the most dangerous thing that can happen when there is an entry of a bacteria. There can be food poisoning, there can be toxic shock syndrome. So this is not caused by the bacteria, but the immune response which is acting against the bacteria. So depending on the type of the organism, depending on the virulence of the organism, depending on the size of the organism, the different kind of immune responses will be activated in our body. So we all know that there are two types of bacteria, gram positive, gram negative, and based on the characteristics of their cell wall, they were named gram positive or gram negative by uh, Christian Gram in 1882, because he developed that gram staining procedure, and this can be seen only under the microscope. So these are the differences between the gram positive and gram negative bacteria. For example, here you can see there is a difference in the membrane structure of both of these types of bacteria. For example, in case of gram-positive bacteria, here you can see peptidoglycan layer is much more thicker, much more multi-layered compared to the gram-negative, where it is very much thin and single layer. Then there is presence of this stichoic acid here in this gram-positive, which is absent in gram-negative bacteria. There is also presence of the periplasmic space in the gram-negative bacteria, which is not that much in case of the gram-positive bacteria. Then there is high LPS or lipopolysaccharide content present in the gram-negative bacteria, which is absent in gram-positive bacteria. Then this physical resistance to physical disruption is high in gram-negative compared to gram-positive. For gram-negative, the inhibition of basic dyes is very much high, and susceptibility to anionic detergent is also high, and resistance to drying is also high. So this gram negative means gram positive means it can retain that color, that crystal violet dye it can retain, and you can see as it as dark violet. So it is gram positive that is retaining the stain, but gram negative bacteria you can it can be decolorized using a counter strain. So it is a gram negative bacteria. So these are the basic differences of gram positive and gram negative bacteria. So as I already told, it can be modes of disease transmission can be various, like it can spread from the food, which is one of the most common source of infection by bacteria, because many times what happens, we eat outside, which is not like food which are not that much hygienic, and there is the case of food poisoning. And also one of the most important thing is water, then fecal contamination, or flies which carry those bacteria, then dust, obviously, then direct contact with the infected person can help us to contract that bacteria. Then animal bites, feces, indirect, some arthropod vectors are there which will carry the bacteria. So via air also respiratory droplets. So various modes of translation are there from where you can contract a bacterial infection. So uh, there can be several factors that lead to the development of bacterial infection and disease. First is the infectivity of an organism, which determines the number of individuals that will be infected compared to the number who are susceptible and exposed. So it depends on the infectivity of an organism, like the bacteria, like how many people it can infect. Like if it is highly infectious, then it can infect a large number of people, and people who are there who are very much susceptible to these kinds of infection, and uh, like people who are exposed to this kind of bacteria, not all will contract the infection, so it depends on the a particular type of pathogen or the infectivity of a particular pathogen, as well as the person who is getting who is susceptible to the particular infection. Second one is the pathogenicity. Pathogenicity is a measure of the potential for an infectious organism to cause disease. So we this is the meaning, like uh, this is the pathogenicity means it is a measure how an infectious organism is going to cause the disease. It is the potential for that particular organism, and there are many ways through which this pathogenic bacteria can evade our immune system and causing disease. So you can understand every day we are getting exposed to millions and millions of bacteria, but still we are not developing that kind of disease every day because our immune system can easily, even if something is entering in our body, any kind of pathogen, then our immune system is able to uh, like protect our body from this pathogen. But there are certain pathogenic bacteria or which has the potential to cause a disease, not in every individual, but to some extent people who are susceptible. As I already told, not all people will be susceptible to the same disease. Not all pathogen can cause disease to everyone. So that measurement of this pathogenicity is also an important thing to develop bacterial infection. And finally is uh, virulence means it's an organism's propensity to cause a disease through properties such as invasiveness and the production of toxins. So there are many bacteria which produces different kinds of toxin which can easily invade the our immune system and can cause a particular disease. So there are several factors that can lead to the development of the bacterial infection and case of disease. So it depends on the organism, depends on the host, depends on the immune system, depends on the production of these toxins or the amount of how it can bypass our immune system. So there are different kinds of host factors which are very critical in determining whether a particular disease will develop following transmission of a bacterial agent. So these factors include genetic makeup, nutritional status, age, duration of exposure, and coexisting illnesses. Obviously, it differs from one person to another. Not a person, a particular family getting exposed to a particular bacteria. Not everyone will have the same kind of uh, like infection. Everyone will have a different kind of infection, and it also depends on the genetic makeup. So there are certain locuses which make a particular person resistant or susceptible. There can, it is known in the, like in studies, different studies are there which has found out that there are certain genetic variation which are present in different kinds of population. Maybe from Asian to American population, there is kind of genetic makeup, or even in a small kind of population in a particular local area, there can be a variation in the genetic makeup which make them more susceptible or more resistant to a particular disease. It can be due to the age because elderly people are more susceptible to different kinds of infection because their immune system are kind of weak compared to the like compared to the adults. Obviously, newborns are also more susceptible to this um, uh, different kinds of infection because their immune system is also weak compared to the adults. So uh, it depends on the person and varies from one person to another. And also duration of exposure to the organism means how much time you're exposed to a particular organism. Obviously, if someone is deliberately getting exposed to a particular organism. For example, I work in a lab where we work with viruses. So if I deliberately go and sit or get like with bare hands, if I take the viruses, obviously I will be exposed to that virus. So it also depends on the duration of exposure to the organism and also with whether that person is having any underlying disease or not. Not only this is true for bacterial infection, but in any kind of disease. For example, we saw during COVID time, like where people who were having underlying illness, especially people with heart diseases, diabetes, they were more susceptible to the COVID. Why? Because the immune system is not able to hold that uh, infection, and so they were more susceptible to the particular virus. So this is one of the important point where people who having underlying disease are more susceptible to this, not only bacteria but other kinds of pathogen infection. So environment also plays a role in host susceptibility. Air pollution as well as chemicals and contaminants also weaken the body's defenses against bacterial infection. Obviously, while we are living in a polluted kind of city or polluted kind of place compared to a place where it is very much less polluted, the immune response will also differ. If there are many studies where they have seen that pollutants, for example, in influenza, there is a study which shows that when there is high pollutant, people are more susceptible to the influenza infection compared to the people who lives in a less polluted area. So it depends on the environment also. So all these factors determine whether a person will get infected by a particular bacteria or any kind of pathogen or not. So there are, apart from the gram positive and gram negative bacteria, like we can divide the bacteria into extracellular bacteria, intracellular bacteria, or facultative intracellular bacteria. So these are the traditional extracellular bacteria, means they can also survive outside the cell, but they are intracellular means they need a particular cell to actually replicate to their genome. Or there are some facultative intracellular bacteria which can uh, survive outside also or as well as inside the cell also. So extracellular bacteria includes this Staphylococcus aureus, S. pyogenes, B. pertussis, this E. coli, H. pylori, etc. All these are known to cause different kinds of diseases, and whereas this obligate intracellular means they only can survive inside the our host system. These are the Lepremurium, Burnetii, Whippley, Staphylococcus. Many are there, and some are able to survive both in like intracellular as well as extracellular phase. So apart from that, there are different kinds of other division like facultative, intracellular. Apart from intracellular, extracellular, there are obligate parasites. There are facultative parasites, which are like you can divide the bacteria into many different forms, and depending on that, there you can classify them. So, as there are two types I showed, there are extracellular as well as there are intracellular bacteria. So, for case of extracellular bacteria, the humoral immune response is the main protective response against these bacteria. So, whenever there is an exposure of this extracellular bacteria, of our host cell to this extracellular bacteria, then it induces the production of antibodies, which are secreted by plasma cells in regional lymph nodes of the submucosa of the respiratory or gastrointestinal tracts. So, whenever these extracellular bacteria are coming inside our body, they are mainly protected, like our body is mainly protected by against them by the antibodies which are produced against them. We all know that antibodies are produced by the plasma cells, plasma B cells, which are traveling in the regional lymph nodes or in any case of in respiratory or gastrointestinal tracts. So, these antibodies can act in several ways to protect the host from invading organisms. For example, mainly IgA is secreted in our saliva. So, these they protect a lot against this extracellular bacteria. Then, this extracellular bacteria also can induce local inflammatory response. Uh, so there are certain toxins which are present in the bacterial cell wall. For example, they are known as endotoxins. They are component of the bacterial cell wall, such as LPS, lipopolysaccharide, which are present on the gram positive. I showed you the sorry, not gram positive, gram negative, and also there are certain toxins which are secreted by this bacteria, which are known as exotoxin. So these different kinds of toxins are secreted by the bacteria, which then activate our immune system and can cause local inflammation. So these toxins are immunogenic toxins, means they can activate our immune system. For example, in case of tetanus, we all know whenever there is a cut with some kind of metal, we go and take this tetanus. Why we take? Because there is a chance that through the cut there is this Clostridium tetani, which is causing the tetanus disease. They can easily enter into that wound. So we go and take the tetanus injection. So this tetanus and diphtheria are disorders caused by exotoxins made by two different bacteria. For example, tetanus is caused by Clostridium tetani and diphtheria is caused by Corynebacterium diphtheria. So both of these can cause these two types of disease, and they are mainly because of the exotoxins which are produced by these bacteria inside our host cell. So when they are producing the exotoxins, there is the immune system gets activated. There is more and more production of cytokines. There is local inflammatory response, and then it can lead to many other kinds of symptoms, and then it can go to a certain extent which is not very much like helpful to our body, and there is chances of many kind of even death also. So uh, for that only we whenever there is a cut, we go and take the tetanus to prevent the entry of this bacteria into our body. So this is an antibody-mediated mechanism for combating infection by our extracellular bacteria. Uh, so here you can see the bacteria is entering through a cut or a wound or a break in our epithelial surface. Here they are producing some toxins. So what happens? This antibodies are secreted by our plasma B cells. It goes and binds to these toxins and they can neutralize the bacterial toxin. both endo as well as exo, they can bind to and they can neutralize these toxins. Then there can be activation of the complement on the bacterial surface, which leads to complement mediated lysis of the bacteria. Here you can see the C3b complement is getting activated. So this there is a lysis of this here bacteria where this complement is binding to the uh, what to say that macrophage is there, which is having this both the receptor for the binding of the Fc receptor for binding of the antibody, as well as receptor C3b receptor, which is binding, and this is forming a complex, and then there is complement mediated lysis. Then there is opsonization as well as phagocytosis. This antibody and the complement product C3b bind to the bacteria, serving as opsonins to increase phagocytosis. Then there can be anaphylotoxins mediated mast cell degranulation, where there is activation of the complement pathway by the antibody. So there are two antibodies C3, two complements C3 and C5a, which are generated by the antibody initiated complement activation, which leads to the mast cell degranulation. Here you can see the granules which are released by the mast cell, and which is helping in vasodilation and extravasation of lymphocytes and neutrophils. So here is the capillary. You can see here the neutrophil, all the cells or the uh, innate immune cells like the macrophage, lymphocyte, neutrophils are traveling. And here you can see when there is an increase in the release of these granules by the mast cell, what happens? There is vasodilation. So it is dilating the capillaries through which these cells can enter into the site of infection. Here you can see through the process of extravasation, it can the lymphocyte, macrophage or neutrophil can enter into this space where it is going and targeting the bacteria. And there are other like complement products which are helping in the chemotaxis of neutrophil and macrophages. So here you can see there are some like like products are there which are helping in the chemotaxis of the neutrophils and macrophages into the site of infection. So this is the one of the way by which our body protects it from the extracellular bacteria. Mostly it is antibody mediated. So in what happens in case of the uh, intracellular bacteria? So this is of the extracellular bacteria by antibody mediated mechanism. And in case of intracellular bacteria, there can be it is it can lead to a response which is a delayed type hypersensitivity response. So here what happens? Some intracellular bacteria is coming. So here you can see this is an antigen presenting cell. So first there is some the cells antigen presenting cell can recognize that some pathogen is coming. So the intracellular it there are peptides which are present on the surface of this intracellular bacteria, and that APCs or antigen presenting cell can recognize those peptides. Then they present these peptides to the naive CD4 cells. Here you can see this is a CD4 cells, and here through the MHC class 2 molecule, this antigen presenting cell is presenting these peptides. These are the chopped or the processed peptides which is presented by the APCs to the CD4 cell. And here what happens? This CD4 T cell is getting primed and it is getting differentiated into Th1 cells, and Here you can see Th1 cells, so T helper cells are they are getting differentiated too, and they are secreting different kinds of cytokines. Here, antigen present in cells can be dendritic cells, macrophages, or Langerhans cells. And here you can see the CD4 Th1 generally and Th17 also they can make, CD2 Th2 as well as CD8 plus cells they can make. So the cytokines are secreted by these Th1 cells where they are acting on their own receptor, making it to proliferate more and more and making new Th1 or other types of cells. This is the sensitization phase where it is recognizing the bacteria and getting sensitized and making more and more number of cells. In the effector phase, after subsequent exposure of sensitized effector T cells to antigen, Th1 cells secrete a variety of cytokines and chemokines including IF and gamma. So here you can see already Th1 cell is sensitized because it is getting exposed to more and more and number of antigen. So it is kind of sensitized. So the response is also fast in this case and it secretes more and more amount of IF and gamma. So this IF and gamma can activate this resting macrophage, making it activated. Here, the macrophage is there, which is also the APC, and it is more and more IF and gamma is secreted. It is activating this resting macrophage, and then it is becoming activated. Then, after a macrophage is activated, obviously it can have a multiple functions. So upon activation it can do phagocytosis. There is an increased amount of MHC class 2 expression, there is increased amount of TNF receptors, there are production of oxygen radical, nitric oxide defensins, and different kinds of chemokines. So here activated macrophage uh, can help in the clearance of the pathogen by different kinds of mechanism. So this is known as a delayed type hypersensitivity response in particularly in case of intracellular bacteria. So why it is called as delayed type hypersensitivity? Because it develops almost 24 to 72 hours after exposure to an antigen when the immune system recognize uh, as something as foreign. So this occurs like much after, like it is a delayed reaction, which is after a considerable time only it is getting activated. So it is known as delayed type of hypersensitivity, and this is more mainly mediated by the T cells rather than the antibodies which are acting against the extracellular bacteria. So extracellular bacteria, the antibodies are protecting, and here in case of this intracellular bacteria mainly the T cells are protecting. But these are all the adaptive immune response which I am talking about. But if in sometimes what happens if there is an infection, sometimes the innate immune response is sufficient to clear off the pathogen via this activation of neutrophils or via this uh, macrophages. So there is no need to activate the uh, adaptive immune response. So it depends on the type of the pathogen again. Like if it is a less virulent pathogen, our immune system can easily clear off it without activating the adaptive immune response. But if it is a highly pathogenic bacteria, then it needs more and more robust intervention, and there is activation of the adaptive immune response. So any question is there regarding this? Okay. So, here also another picture you can see, which is showing the different kinds of. Uh, can you see my slide now? Okay. So, so this is a so this is a overall picture. What happens when a pathogenic bacteria is entering into our body? So, once pathogens are recognized by the extracellular endosomal receptors, the host cell uses bacterial compartmentalization, oxidative and nutrient stress, antimicrobial peptides, lysosome-mediated degradation, autophagy, inflammasome activation, and pyroptosis to kill the pathogen. So, there are different kinds of processes through which our body can kill the pathogen. So, for example, this pathogenic bacteria is entering. It can lead to it can our body, our cells can compartmentalize it. Like it can form a compartment outside the bacteria where the bacteria can enter into this compartment. There can be production of ROS and antimicrobial peptides. Then there can be activation of the inflammasome. There can be regulation of the inflammasome. There can be processes such as autophagy, where there can be production of the autophagosome, in which will now cleave the bacteria into small pieces. Then there can be pyroptosis, there will be killing of the bacteria. There will be different kinds of uh, like modulation of the vesicle traffic. Like our host system will prevent it to and prevent the bacteria to enter into the host into another other cells. So it will restrict the vesicle traffic. There can be escape also. These bacteria can escape obviously from the vacuole. There can be reorganization of the cytoskeleton. Then this innate immune activation, controlling of the innate immune activation. Then fusion with the host vesicular compartments, inhibition of lysosome fusion, proteasome degradation of host protein. So uh, our cells are also trying to uh, kill the bacteria, but the bacteria can also try to uh, evade our responses inside the host cell. So it will try different kinds of mechanism to delay the different processes inside happening inside the cell. Like example, it can degrade the host proteins. it can inhibit lysosome fusion, it can fuse with the host vesicular compartment, it can control our immune system, it can reprogram our host gene expression and chromatin remodeling. So it controls different kinds of processes in our body so that our system also cannot go and remove the bacteria from the cell. So, some intracellular pathogens can control the signaling pathways activated by host receptors, interact with the endocytic pathway, escape from the phagosome, inhibit fusion with the lysosomes, manipulate vesicular trafficking, and avoid autophagosome degradation and inflammasome activation. So, you, the bacteria also tries to evade all these processes, and so that it can also uh, multiply inside the cell and can infect other cells, because it also wants to survive in our body while our body also tries to remove it from the system. But there are various ways through which various bacteria, for example, one of the most common is this antibiotic resistance. This bacteria will secrete this antibiotic resistance gene so that antibiotic also won't work and it will be able to survive in our body. So here you can see an example how different kinds of bacteria are evading the responses. So first bacteria is internalized by the host cell. So you can see this is forming a compartment, is a vacuole through which bacteria is entering. So once inside the cell, different pathogens follow alternative pathways of intracellular trafficking to establish a replication niche. So depending on the type of pathogen, depending on the type of bacteria, it will follow different kinds of pathways. For example, there are certain bacteria which can reside in specialized vesicles that are termed as vacuoles or phagosomes and can manipulate the host cell molecular motors to induce vacuolar and vesicular movement along microtubules. Here you can see some, these are the example includes this salmonella. What salmonella is doing? Salmonella is residing in these specialized vacuoles, which are known as phagosomes or vacuoles, and they can help uh, in the they can manipulate our host cell system to induce the vacuolar and vesicular movement along the microtubules. Here you can see along the microtubules they help this, they are reorganizing, they are so smart that they are reorganizing the whole cytoskeleton and they are these microtubules and they are moving along with it inside the vacuoles. So we all know that there are two ends of the microtubules, one minus end and one is a plus end. So in non-polarized cells, microtubules are organized with minus ends gathered at the microtubule organizing center and plus ends extending towards the cell periphery. So these bacteria can control that whole cytoskeleton, like whole cytoskeleton, and they can reorganize this according to their own will. So there are other bacteria like vacuolar bacteria. They can also interact with different host cell compartments of the endocytic pathway, such as endosomes, lysosome, and can replicate in host cell compartment such as the ER. So here you can see ER is there, Golgi is there. So they can enter into that easily. Here you can see the bacteria can enter easily into the ER and they can replicate inside this different kinds of uh, membrane, like these different kinds of organelles according to their own wish. It, for example, includes Brucella and Legionella. Some other bacteria are there which escape from vacuoles to remain into the cytosol. Like this, you can see escape from vacuole. These are the example. For example, Listeria, Shigella, Rickettsia, and Francisella. They will escape from the vacuole because they want to stay in the cytosol and then promote actin-mediated motility. So, with the help of actin, they also will travel to different places. Here it includes this Listeria, Barker, Holderia, Mycobacterium, Marinum, Shigella, Rickettsia. etc. So you can see that different kinds of pathogens, different kinds of bacteria are using different kinds of mechanism inside the host cell to survive. So other examples are when there is a here you can see lysosome. So the bacteria will want to avoid the lysosome because there are lytic enzymes present inside this lysosome which will kill the bacteria. So they want to escape from these lysosomes also. Uh, so to survive, they what do almost all intracellular pathogens inhibit or delay lysosome fusion. So they will try to delay this fusion of the lysosome because otherwise these lytic enzymes will kill this bacteria. So example includes this Coxiella, then there is Salmonella, which is preventing the fusion. Then here you can see Mycobacterium spp, which this is early endosome and this is recycling endosome. So if there is a fusion of this endosome with the host cell membrane, or if there is a fusion of this vacuole also, then this bacteria will go inside and there will be a fusion with the lysosome also. So all these bacteria also wants to avoid that, and they will uh, they prevent this fusion with the lysosome by different kinds of mechanisms. So you can understand more than the host cell itself, the bacteria are more smart because they are evading the immune system so well. So so these are the primary steps in most bacterial infection. First, they will attach to the host cell. There will be proliferation. There will be invasion of the host tissue, and there will be toxin-induced damage to the host cell. So, host defense can act at each of these steps, and many bacteria, as I told, have evolved ways to circumvent most of the them. So, first, at every step, our like there are some whenever there is a the bacteria which is trying to attach to the host cell, our system will try to remove it. Then, if it is getting attached and entering into the cell, then it wants to get proliferate. So, it will try different kinds of strategies to evade the host response. As I showed you earlier, like it will try to bypass the system, or it will take help of the our own system. Like example that microtubules or actin to move into different places, so that it can proliferate, because one of the main agenda of these all pathogens is to survive and proliferation and make more and more copies of themselves to infect more and more cells or more and more persons so that they can survive. Then they will invade the host tissue. Then there can be systemic invasion also. Not only the host tissue, but when there is a systemic invasion, it might lead to different kind like septic shock or any kind of this shock syndrome diseases. So, and there can be toxin induced damage to host cells. So these are the different kinds of uh, at every step our immune system is also working, but at every step the bacteria also has different kinds of strategies to circumvent the uh, strategies our immune system are trying to imply. So this is one table which is there is any question? Okay. So these are the different strategies by which bacteria can evade our response. For example, attachment to host cells. If some bacteria is trying to attach to the host cells, there is IgA antibodies, as I told, which block this attachment by secretory IgA antibodies. However, there are many bacteria which has learned to bypass the mechanism. They will secrete some proteases themselves that will cleave the secretory IgA. For example, Neisseria, Neisseria meningitidis, the Neisseria gonorrhoeae, Haemophilus influenzae. So they have certain proteins or proteases that will cleave these IgA dimers, and they can evade the immune system. Then certain bacteria are there which prevents the phagocytosis process. They, while proliferating, if certain antibody and C3b mediated opsonization will be there, as I showed you earlier, so they will produce some surface structure. For example, they will produce some polysaccharide capsule, then M protein, then fibrin coat. So that will cover the surface of this bacteria, and it will inhibit the phagocytic cells. So if there is a presence of any coat or capsule, then the immune system will not be able to recognize these pathogens because the antigens are kind of uh, shaded, or it is kind of hidden from the immune system. So they will escape this phagocytosis process. Then many have also developed certain strategies for surviving within the phagocytic cells. Then there is induction of apoptosis in macrophages. For example, by Shigella flexneri, they will induce apoptosis in macrophage cells. So instead of phagocytosis, the macrophage will itself die out of apoptosis. So there is also complement mediated lysis and localized inflammatory response in certain cases. Generalized resistance of gram positive bacteria. Some gram positive bacteria are resistant to these processes, and there is some, in case of some gram negative bacteria, there is production of this long side chain in the LPS, cell wall LPS, which will prevent the formation and insertion of the membrane attack complex. So you can understand this bacteria also tries to evolve according to the immune system. Like if there is a complement mediated lysis, they will produce this long chain block side chain so that the membrane attack complex cannot be formed on the surface of this bacteria. So in case of some, for example, in Pseudomonas, what they do, in case of the Pseudomonas, when they're invading the host tissue, there will be antibody-mediated agglutination. But these Pseudomonas, what they do, they will secrete elastase, which will inactivate C3a and C5a complements. And so this elastase will break down these complements, and they will survive the evasion mechanism. Then there is toxin-induced damage to host cells. When there is toxin-induced damage to host cells, there will be neutralization of this toxin by different antibodies. However, some bacteria will secrete hyaluronidase, which will enhance this bacterial invasiveness, and there will be no neutralization of the toxin by the antibody. So these are some of the examples by which the bacteria can evade the immune system. As I told the example, some bacteria express molecules that enhance their ability to attach to host cells. A number of gram-negative bacteria I showed in the picture have pili, or they are long hair-like projections which enable them to attach to the membrane of the intestinal or urogenital tract. Other bacteria such as Bordetella pertussis, which is one of the cause of whooping cough, secrete adhesion molecules that help the bacteria to ciliated epithelial cells of the upper respiratory tract. Then secretory IgA specific for such bacterial structure can block the attachment of these on the surface and one of the most host defense against many of these bacterial strains. But however, as I already showed, some bacteria such as Neisseria that cause gonorrhea as well as meningitis evade the IgA response by secreting some proteases that will cleave the dimer. IgA needs to make a dimer to have its action, but these bacteria will cleave the dimer at the hinge region where they are joining, and thus resulting the AB and FC fragments having a shortened half-life in mucus secretion and are not able to agglutinate microorganisms. So different kinds of bacteria having different strategies. These are some of the other examples. For example, by which they will bypass the system. So the antibody responses made by the host can be evaded by some bacteria that undergo frequent changes in their surface antigens. For example, as I already told, with Neisseria gonorrhea, they bypass this IgA antibody. They have this pili on its surface, which is having a highly variable structure, and it can be generated by gene rearrangement in the coding sequence. So there can be a permutation combination of the gene rearrangement and it will synthesize new and newer version of the pili. So this process generates enormous antigenic variation, which may contribute to the pathogenicity of this N gonorrhea by increasing the likelihood that expressed pili will go undetected by antibody, allowing them to bind firmly to epithelial cells and avoid neutralization by IgA. So as you can understand, if it is generating a highly variable structure like different kinds of pili, if it is generating, then not every time our immune system will be able to recognize that particular bacteria. So what it will do, every time it will be a new kind of bacteria because the antigen itself, surface antigen itself, if it is changing, so the IgA also cannot go and bind to the different kinds of pili that it is expressing, and then it can avoid neutralization by the IgA and can escape the immune system. Bacteria may also possess surface structures that inhibit phagocytosis. For example, one example is Streptococcus pneumoniae, whose polysaccharide capsule prevents phagocytosis very effectively. For example, there are 84 types of S. pneumoniae which differ from one another by distinct capsular polysaccharide. The host only produces antibody against the infecting serotype, not the others. For example, when suppose number one serotype is infecting you, then our host system will recognize it and will produce antibody against it. But when number two serotype is infecting you in the second time, then what happens? Our host cannot recognize it because the whole surface is changed. So it will again take time to produce that antibody. If it is similar, only one type of serotype is there, that means already some memory will be there in the second case. So it can go, that antibody will be produced in a flash of moment and it can go and bind to that particular serotype. So it is just delaying the immune response every time. So this antibody, which is protecting against reinfection with the same serotype, but will not protect infection by most of the other 83 serotypes. So one antibody can detect only one serotype, but so many variations are there in the surface antigen that it cannot detect the infection by other serotype. In this way, genetic variants of S. pneumoniae can cause disease many times in the same individual. So you can understand like 84 serotypes means how many types of serotypes they are producing. Like like one person, if they are getting infected with 84 serotypes, that means 84 times the antibodies will be generated. Otherwise, if there was only one serotype, then one antibody can detect reinfection, but not the infection of the new serotype. So Apart from this, some pathogenic staphylococci are also able to uh, assemble a protective coat from host blood proteins. These bacteria secrete an enzyme coagulase that precipitates host-generated fibrin, making a coat around them and shielding them from phagocytic cells. So another master stroke by this bacteria is using the host system to protect it. These I am talking in case of streptococcus or in gonorrhea, they will form their own structure. But this what staphylococcus is doing, they will synthesize one enzyme coagulase which will precipitate the fibrin generated by the host. So from the blood proteins, fibrin is a blood protein help which helps in the clotting procedure. They will secrete that enzyme which will precipitate the host fibrin, and that fibrin will make a coat around them and shielding them from the phagocytic cells. So our phagocytic cells cannot recognize the bacteria because it is thinking like this is their own system, because fibrin is a host protein. So this is one of the best and the master stroke, are very much clever these bacteria are, that they are using the host protein itself so that the immune system cannot recognize this bacteria. So so mechanism for interfering with the complement system also help other bacteria survive. For example, in some gram-negative bacteria, long side chains, as I showed, these long side chains will be made of lipid moiety of the cell wall core polysaccharide help in resisting complement-mediated lysis. Pseudomonas secretes an enzyme elastase that inactivates both the C3-5 and C5a anaphylatoxins, thereby diminishing the localized inflammatory reaction. So the Pseudomonas will secrete an enzyme elastase which will inactivate the C3A C5 complements, thus diminishing the localized inflammatory reaction. And in case of some gram-negative bacteria, they will synthesize a lipid chain so that there is no formation of the membrane attack complex. Then, a number of bacteria escape host defense mechanism through their ability to survive within the phagocytic cells. For example, Listeria monocytogenes escape from the phagolysosome to the cytoplasm, a favorable environment for their growth. Other bacteria, such as Mycobacterium genus, block lysosomal fusion with the phagolysosome or resist the oxidative attack, allowing them to remain and replicate in endosomal vesicles. So these are the different strategies opted by different kinds of bacteria, and they will do different kinds. So different kinds of strategies by this bacteria to evade the immune response. So here you can see Neisseria gonorrhoeae. This is the bacteria, and here you can see these are the pili, which is extending from the gonococcal surface and which is helping in the attachment to the target cells. And as I already told, this Neisseria gonorrhoeae will have number of pili in its surface generated by gene rearrangement in the coding sequence. Thus, not every time it will be recognized by IgA. If there is some kind of reinfection only, then that will be recognized by the IgA. Now we come to the Mycobacterium tuberculosis, which is again one of the bacteria, which is a famous bacteria everyone knows about, because it has caused so many deaths. Not now, because some treatments are there which is preventing, but still it is again one of the bacteria which is having multiple drug resistance. So you can understand, like, this is another smart bacteria which is escaping the immune system. So, Mycobacterium tuberculosis was first described as the causative agent of tuberculosis by Robert Koch in 1882 on 24th March, which is known as the uh, TB Day now. So, it is a slow-growing, chemo-organotrophic, non-motile, non-spore-forming aerobic bacillus. This is the structure of the Mycobacterium tuberculosis. It is having this capsule, this cytoplasm is there, DNA is there, pili is there, plasma membrane. This is the mesosome, ribosome, plasmid, flagellum is there. So it is a typical bacteria structure which I showed you earlier. So it is a very slow growing chemo-organotrophic, non-motile. It is not motile. It does not form any spore, and it is aerobic bacillus. And aerobic bacillus means it requires air, and chemoorganotrophic means it requires energy by the oxidation of the organic compounds of the reduced organic compounds. It will oxidize and gain its energy. Under laboratory condition at 37 degree, it doubles every 24 hours, taking approximately 3 weeks to form buff-colored rough colonies on agar plates. It is visualized by the Zeal-Nielsen acid-fast stain, whereby its thick waxy cell wall retains carbon fusion stain in the case of acid-alcohol washes. And it is like it can be visualized only on the microscope, obviously, and however, it will form in agar plates. We plate the bacteria to see colonies. You can see those colonies in your naked eye because colony won't have one bacteria. It will have a large number of bacteria sticking together, and it can be visualized by different kinds of stain, acid fast stain, and carbon fusion stain. And there are different kinds of biochemical tests, including positive niacin production and the ability to reduce nitrate, which helps it to differentiate it from the other mycobacteria. So mycobacteria has different kinds of species, and out of that, Bacterium tuberculosis is one of the species. And since it is causing tuberculosis, so much research is going on still now, because especially on drug-resistant TBs, because it is still not eradicated yet. So tuberculosis is a disease which is caused by Mycobacterium tuberculosis. It is remains a significant global health problem despite advances in diagnosis and treatment. However, approximately 10 million new cases of TB are diagnosed each year, with over a million deaths attributed to the disease. So you can understand 10 million new cases every year it is diagnosed. So how much dreadful it is, deadly disease it is, despite having this much medical assistance. TB is particularly prevalent in developing countries where co-infection with human immunodeficiency virus and poor living conditions are common. Yes, it is mostly prevalent in people who is having HIV because they are immunosuppressive, they are immunodeficient, and where people where poor living conditions are there. So it mostly is occurring in people with underlying diseases or who are on immunosuppressive drugs or uh, deficient. So one of the most um, significant challenge is the emergence of the drug resistant, as I already told, strains of Mycobacterium tuberculosis, which is posing a significant challenge to global TB control efforts. There are many drugs, drug sensitive TB also is there, but drug resistant TB is also there. So there are different drugs, for example, isoniazid, rifampicin, pyranizumab, ethambutol, but treatment can be lengthy and costly, with patients often requiring 6 months of therapy. Moreover, the emergence of multi-drug resistant TB and extensively drug resistant TB has complicated the treatment landscape. So there are different uh, subdivision of resistance in TB. For example, there can be multiple drug resistant, like it will be resistant to many drugs. There can be extensively drug resistance means it is too much like nothing can be done against this because no multiple means at least you can do you can treat something to it, but not this extensively drug resistant TBs are very, very difficult to eradicate because they have been resistant to drugs, and there is you cannot undo the infection. So too many strains of TB are there, uh, and which has been developed over the years, and they are all gradually becoming drug resistant. So this is the way in which it infects a host. First, it enters into the respiratory tract and lungs of the host. Then it incites an innate immune response and is engulfed by pivotal immune cells such as macrophage and dendritic cells. Here you can see it can spread from the one person to another via air also. So there is first it infects the respiratory tract, then it is engulfed by these macrophage and dendritic cells. And as I already told, the MTB can replicate within these cells, and more immune cells are recruited to the site of infection. Here you can see primary infection, active tuberculosis, and a new cycle of infection. So first it is invading the respiratory tract and the lungs, exposure to multiple host cell receptors. Here these are the cells which come into the play. Uh, One of the most, what to say, important thing or uh, flavored way by which TB infects is the formation of a granuloma. That is, here you can see it is forming a granuloma. So granuloma means it is where these cells are also there, all the immune cells are also present, and the TB is residing in that cells. So whenever there is a break of this granuloma, this uh, infection can go and infect the people. So first it is invading the respiratory tract and lungs, then the innate immune cells are getting activated, it is going and spreading, and there is intracellular replication, there is production of the proteolytic enzymes and cytokines, there is uh, activation of the adaptive immunity. Then there is recruitment of the immune cell. Either it can lead to complete eradication, or in a vascularized structure, if these immune cells, along with the infection and infect like the bacteria, can reside in a highly vascularized cell. So this granuloma is forming means it can stay in this phase for a long time. So this granuloma are composed of foam cells derived from macrophages, the necrotic immune cells, um, and whenever there is a rupture of this granuloma, they will release the bacteria, resulting in the development of active TB. So here you can see this bacteria will be released. So when this MTB bacteria released as infectious aerosol droplets, there will be a new cycle of infection. So there can be two stages of TB. One is active TB and one is latent TB. Latent TB means where the bacteria is not doing anything. It is in a uh, the particular bacteria is inside your body and it is like they are sleeping kind of means inside the granuloma they are well and fine. They are not doing any immune response. They are not activating any immune response. They are just sleeping. But whenever there is a breaking of this granuloma, then it makes a active TB. So that when this bacteria is released, then it is recognized by the immune system. Then there will be uh, many kinds of reactions that are going on in the cell. So there are two types. One is active TB and another is the latent TB. So in case of the active TB, in the case of the latent TB, the TB bacteria will reside into your cell and you cannot understand whether you are infected or not. That is one of the most dangerous thing, that you are harboring a bacteria and you cannot understand that whether you are having an infection or not. So it will live peacefully inside your body and won't grow in the body. It will just sleep. It won't make you feel sick or you are not having any symptom. You cannot spread also it from one person to another. So uh, you are just fine with it, living with the TB like inside your body. Suppose in the lungs itself, it can go and accumulate and is staying. No, it is not affecting, it is not doing any harm to you. But suddenly one day, what happens? This granuloma is breaking. So what happens? The bacteria is releasing, infecting your whole lungs. So this can happen anytime, and this can advance to a active TB. So this when there is active TB, it can grow in the body, it can make you feel sick. There can be plethora of symptoms. It can spread from one person to another, and if not treated, it can lead to a disease, dreadful disease. So you can understand like how this active versus latent TB is creating a kind of havoc in your body, and people sometimes cannot understand whether they're having TB or not. Like randomly, doctor if suggests whether to see if someone is having TB, they will do tests, but they won't have any idea like they are having TB or not. But they will, after the test, they are seeing that they are having latent TB. So this is one of the like most bad thing, like having a latent TB inside your system, not able to clear it, but just living with it. So here are the two different phases, as I already told, and both phases can be treated. The medicine, first, when TB germs are entering in your body, they cause latent TB. Without treatment, latent TB can become active TB disease. So it is advisable to know that whether you are having latent TB or not, because if it is becoming active and if it is a multi-drug resistant or a drug resistant TB, it is very difficult to clear up the infection. So in phase 1 latent TB, it is asleep in your body. This phase can last very long time, even many years. Many years you can stay with like that without any harm in your body. You don't look or feel sick. Your chest X-ray is usually normal, and you cannot spread the TB to the other people. Usually treated by taking one medicine for 9 months. Phase 2, which is active TB disease. TB germs are active and spreading. They are damaging tissue in your body. You usually feel sick. Your doctor will do special tests to find where TB is harming your body. If your TB is in your lungs, you can spread them to other people by coughing, sneezing, talking, or singing. You can treat them by taking 3 or 4 medicines for at least 6 months. So you can understand if it is latent TB, it is not that much harmful, but the latent TB has the potential to become active. So it is better to treat the disease in its latent phase itself before it is getting active. So there are various ways through which the doctor can give you tests. For example, one is the tuberculosis skin test or the Montauk's test we call, where if they will give a purified protein derivative, tuberculin protein, which they will inject in your system, and they will see whether there is any kind of what to say, any kind of reaction to that particular protein, because if there is Mycobacterium tuberculosis, then there will be positive. The test will be positive because your immune cells can recognize this antigen because they already have recognized your previous antigen from the Mycobacterium, and there will be uh, inflammation at that particular area where they have put the antigen. And if the test is negative, that means you are not uh, been infected with the TB. So particularly this skin reaction develops after 48 to 72 hours, and there is if there is a like inflammation in that area, it is positive. If there is a no inflammation in that area, it is a negative. So it is useful in diagnosis of tuberculosis. However, it is not sufficient for interpreting the exact result, and you need more like clinical signs and symptoms will be needed and other things will be needed, because sometimes one antigen can also cause like sometimes some reaction can happen with a new antigen also. But this is one of the most tests which is done in the hospitals. So this dermally, this purified protein derivative is injected, and it will if your body has recognized the antigen previously, so there will be activation of the T cells, and then this it reacts with the sensitized T cells and produce hypersensitivity reaction. So 24 to 72 hours the doctor will come and ask see the site to see if there is any inflammation, and in that particular place there will be local hardening, and there will be swelling, there will be redness around the injected area, and there will be erythema, like redness will be there. And so the mode of induration, it is known as like, if the site is swelled and there is local hardening, that diameter of the induration they will measure, and it is directly proportional to the degree of sensitization. So this is a common test, intradermal tuberculin test, that is done in the hospitals. So apart from this, there is another test which is the IFN gamma release assay. So in this case, what they are doing, like when there is an infection, so this is also a whole blood test that can help in the diagnosing in the uh, Mycobacterium tuberculosis infection, like whether the person has been infected with it or not, and it works by measuring the body's immune response to the TB bacteria. So it is a kind of blood test which is done. So it also measures the immune reactivity to M tuberculosis. So most of the people who are infected by this M tuberculosis, their white blood cells will release interferon gamma when you are mixed with some antigens. For example, for the M tuberculosis antigen, and then if that particular person has been infected, then that antigens will be interacting with this IFN gamma, and your blood will be taken, then there will be mixed with antigen as well as control, And when there is a mixture, then there will be uh, it is a positive test, like interferon gamma has been secreted from your body, and there is like infection in your body. And there is here you can see, for example, here antigen presenting cell or APCs will encounter one antigen. Presenting cell ingests, then digests antigen. It will digest. Then it is lead to the incubation. Then presenting cell prevents presents antigen to specific T cell. T cells will get activated and secrete IFN gamma. So after that, uh, antigen specific cytokine production, like this IFN gamma will be secreted. Then you have to see quantify the IFN gamma. If the antigen is TB specific, only TB specific T cells will activate and secrete IFN gamma. Here you can see TB specific peptide antigens are kept, and there will be production of the IFN gamma, and using different procedures like ELISA as well as plasma, or you can do a ELISpot assay to identify how much cytokines are produced. So this is a IFN gamma release basically, which is uh, detecting whether you are having TB or not, whether your cells are being sensitized to produce IFN gamma or not. So these are the two tests which are done against TB, and a few other tests are also there, uh, which also are conducted, but mainly these two other tests which are done. So apart from this, there are, as I told, different kinds of drugs are there, which are there for TB, but most of the TB nowadays are drug resistant. So there will be oral drugs, there can be injections, there can be other like there can be drugs where they're combinedly given so that more effect is there. So it is one of the concern that many and many drug resistant strains are coming up these days, which has to be which so research is going on to reduce the amount of this drug resistant TBs. Some example of the TB drugs are mainly isoniazid, rifampicin, ethambutol, streptomycin, etc. But uh, all these many bacteria are getting resistant against all these drugs. So it is important to make more and more new drugs which can be active against the TB. So any question is there? Okay, so two doubts were there. Uh, for example, someone, Arun, who has asked the question, like he had a doubt in the answer key of assignment 9, question number 2, and he had a doubt like the question was which of the following is not a function of type 1 and type 3 interferon, and his doubt was the answer given is cleaves procaspase into active enzyme, but can the option virus specific immunity be an answer? Kindly respond. So the question was which of the following is not a function of type 1 and type 3 interferons. Not a function here, they have told. It is not like which of the following is a function. So the options were killing of infected cells, makes healthy cells resistant, depress cellular synthesis pathway, prepare or activate professional immune cells for development of virus, uh, specific immunity, then cleaves procaspase into active enzyme. So here it is not, the question is not that which of the following is a function of type 1 and type 3 interferon. It is not a function of type 1 and type 3. So we all know like type 1 and type 3 interferon can help in killing of the infected cell. It can make healthy cells resistant. It can also cellular synthesis pathway. It can reduce or depress. It can also help in the development of the functional immune cells. It also helps in virus specific immunity. The only option which is not correct in this case is cleaves procaspase into active enzyme. This is not done by type 1 and type 3 interferon. So the answer will be the sixth option, which is cleaves procaspase into active enzyme. So I don't know why he thought like virus specific immunity, because it is one of the most important function of type 1 and type 3 interferon, where it is actually helping in the synthesis of type 1 and type 3 interferon is there whenever there is a virus infection, and it is helping in clearing of the virus from the system. So the sixth option is the only option which is not a function of type 1 and type 3 interferon. So I could not understand what he wanted to say, but it is, I maybe it is like he understood that it is one of the function of type 1 and type 3 interferon, but it is not a function of type 1 and type 3 interferon. So the answer will be the last option. That is the cleaving procaspase into active enzyme. Another question I don't couldn't understand what she asked, Priya. Like she has a question regarding this mycobacterium uh, slide microbacterium video, but in the end, sir has not told anything specific, so I couldn't understand. So anyway, I will reply her back regarding her question. So these are the two doubts this week which were posted in the portal. So anyone of you has any doubt apart from this, or any kind of doubt regarding the lecture or assignment? Okay, so anyone, I suppose no one is having any doubt. So if any doubt is there, you can mail or you can post in the portal, or next week will be the last class, doubt clearing class. If anyone is having any doubt, they can join and ask regarding any kind of doubt. So that's all for today. Thank you.